Okay, so bacteria in nitrogen fixation N2 reacts. So here we could have bacteria that are involved in nitrogen fixation is what? These are prokaryotes, as we all know. They're simple molecule. Are prokaryotes. They're very simple. They're prokaryotes. They what? They transform nitrogen gas into ammonia or nitrates that are used in plants. Or nitrates that are used in plants. Absorption in soil. In plants. Absorption in soil. Like the enzyme that uses these bacteria to destroy the presence of oxygen. Suggest why destruction of this enzyme. The diagram. Okay, so here's a diagram. Oh, because the structure looks kind of like plants, because here we have chlorophyll. So like you know how plants photosynthesize, instead of photosynthesize they do the opposite. They do the same thing with bacteria. So what they have the structure, the chlorophyll like structure, and the Calvin cycle within it. Photosynthesis, oxygen produced, released. So basically, for this structure, we could see here it looks contains exactly chlorophyll, so we know it photosynthesized. Well, not exactly photosynthesized, but like nitrogen synthesized, you know. So it's gonna produce what? It's gonna produce nitrogen as like it's gonna use light to produce nitrogen to use for nitrate fixation. So like photosynthesis alike. One adaptation the bacteria may have developed in order to prevent, well, what we have thick wall, we have minimal movements of O2, thick walls. Identify each of parts. Seed drop pea plant. Seed drop non-endospermic. Endospermic. So the outside is the layer. So we have the embryo. The entire thing is the embryo. Oh, not the embryo, the coat of the seed. The entire thing is the embryo. Seed coat. We have cotyledons. F is cotyledons here. D is what? D is plumula, and E is Cecal, plumule, radical, cotyledones. Yeah. Name the part of the cell that contains the genes, chromosomes. The chromosomes. Define each of the following term: the gene, protein, coded, more expressive allele. Gene made of DNA, pass on. Dominant allele, its version for gene, expressive allele, version of a gene. A homozygous okay. So we know that this is RR, wrinkled surface. So half of it is wrinkled. So the other half must be not wrinkled. It could be our homozygous genotype. So if this is homozygous genotype, H is R, then this should be R. Okay. The type tree frog of the species is either gray or green in color. The following are both true for gray and green frogs. They eat insects, leaves on the ground, small pools. Natural selection? Wait, no, not variation. Herbivore. Species that only consumes plant-based structure, like vegans. Trophic level of each tree frogs. It eats insects, so like secondary natural selection. Not sure about A. We could check. Discontinuous. Oh, discontinuous variation. That is the knowledge of the process of natural selection in this degree. Okay, it's the process of natural selection, which evolution based on environment, which makes gray frogs commonly found in trees with gray bark. Gray frogs develop what gray color to blend in, to camouflage for survival. It's always about survival. Blend in, camouflage. Green frogs develop green colors, camouflaging. What else? Swamp and mesh. Variation, varieties, mutation, camouflage, survive, to breed, to pass on. What is the cause of mutation? Mutation always, it's spontaneous. Spontaneous. It's random. And the genes would be passed on. Okay. Name each process from K to N. Fossil fuel. So K would be combustion. L, living plants. D, decomposition. What M is carbon dioxide. What photosynthesis. Dead plants and animals. No, living plants. L is not decomposition. This one is decomposition. Respiration. Yeah, respiration. Suggest an explanation pattern change in carbon dioxide is shown the graph. So we say what we see. Let's say what we see. Carbon concentration, length of daylight at location X. Medium, long, medium, short. Sure. Oh, okay. So low CO2 concentration with high light intensity. This is photosynthesis. This is making chlorophyll with chloroplast. Chloroplasts involved. What else? Daylight allocation. Okay, we could say what sept has the lowest CO2 concentration, gradually increases after September. Yep, falls more. We just say what we see. 1 year location X. So we just have to swap back. So instead of what going down, so we just go from here. It goes plateau, it goes there, and short, it goes down. So yeah. Okay, so section B, we could see the blood vessel. If I'm sitting here, so it's gonna be my structure Q is my left ventricle. Left ventricle. Yeah, what does the left ventricle do? The left ventricle. It pumps oxygenated blood to the body. What else? It pumps oxygenated blood to the body. What else? There's one more. It is the main chamber of your heart. Yeah, structure R. So I'm gonna teach you a way to remember all the valves in your body. Do this trick. Try me pa. Me and my friends call it the try me pa technique. Try is here. Try me p a. Try me pa. So try stands for tricuspid, me stands for mitral, P stands for pulmonary, and A stands for atrium. Atrium ventricle. Atrial ventricle. A ventricle. So structure R is the what? The tricuspid ventricle. It separates it separates separate right atrium and right ventricle, prevents blood backflow of the blood, prevents backflow of the blood, or mixing prevents deoxygenated blood and oxygen blood mixed together, prevents the oxygenated blood and oxygenated blood from mixing, so it prevents a lot of backflows and mixing. Blood vessels. P is the pulmonary vein carries deoxygenated blood. It's the pulmonary vein. I think it's the pulmonary vein. It carries pulmonary vein. It transfers oxygenated blood to the left atria. This is probably the first time your vein carries any like oxygenated blood ever in your body. Not from the lungs. It has to get it from the lungs. The rest of your veins carry deoxygenated blood. Okay. The coordinate response, okay. Negative feedback. Coordinate response. What do you call that one thing? Oh, I can't remember the name. Reflex. Is it reflex? Yeah. So it goes through what? It goes from effector, we get a line, effector to sensory to sensory, relay, motor, and to receptors. It goes through synapses, the neurotransmitter pass from dendrites to synapses, goes through the CNS, which is spinal cord, in the brain. Yeah, we got it. Impulse, synapse, receptor, sensory, relay, CNS, great motor effector. Yeah, it's an impulse. So we pretty much got all 7 marked from just naming the sequence. Effector, sensory, relay, motor, receptors. It goes through synapse. It passes from dendrite to synapse. It goes through the CNS, and it is an impulse. Yeah. So that's how you get 7 mark on this one. Damage produce spinal cord host relay relay neuron. What else? It is part of the CNS central this, and it when it hosts relay neuron and it's got damaged, then none of the relay neurons cannot work. Can work. Either question 8 or question 9. Describe the process of the good outside plant absorb outside. Water molecules increase the state of the loss. Okay, so for this section, we're gonna answer both question and in your and you could choose in the real exam. Remove molecules from the blood from person is air fluids. Okay, it goes through CO2. Well, it's also called deoxygenated blood passes in the capillaries. That's one. Goes through alveoli, which are oxygenated alveoli, which are oxygenated, and it does something called diffusion of CO2 into the alveoli and O2 from alveoli into capillaries, and then from there it goes from alveoli to the bronchitis, to the bronchi, to the trachea and nose. I cut down a bit of process, but we outlined the basic idea. Let's see. Yep, we got diffusion, hydrogenated, high to low, dissolved mucus, alveoli. So this is called exploration.